Good morning, everyone. I, Dr. Atrey Shahana, welcome you all to this one-day national webinar on understanding life in light of modern research, organized by Department of Zoology in collaboration with IQAC, Michael Murusudhan Memorial College, Durgapur. Now, at the very beginning, I want to welcome the person without whose constant support and inspiration this webinar would not have been possible. Our Honorable Principal Sir, Dr. G. M. Helaluddin. Now, I request our Principal Sir to please share a few words with us. Over to you, sir.
Thank you, Dr. Sahana.
Thank you, sir. Thank you.
Actually, I have some personal work. Because of that, I will not take much time. 2-3 minutes, I will take your time. So, bear with you. So, it's better to speak in English because um, uh, time is very short. Okay. So, very good morning and a warm welcome to all participants, uh, students, researchers, faculty, resource persons, and others in this national webinar. And today we are going to discuss on recent trends, development of biological research, and its impact on our life. And this is the theme of our seminar, online seminar today. Basically, biological science is the study of nature and microorganisms which have strong role in our life and environment, as these are ultimate rulers of the living world. As we witnessed in the recent effect of coronavirus and its pandemic throughout the entire globe, and an unpredictable, unprecedented situation is continuing for last couple of months, which has absolutely disrupted our normal life, normal activities, and everything which we are experiencing today. Research works are going on in almost all countries in respect to development of vaccine, and number two, vaccine, medicines, preventive medicines, and number two, the preventive measures, how we can handle this disease. But still now, no successful achievement is established in this field. But we badly need to overcome the situation, and hope our subject experts will elaborate in detail in this field, so I'll not go in detail. So thank you so very much and wish grand success of this effort of Zoology Department, Dr. Atri Shahana, and other faculty and staff, their team. So thank you very much. Okay, that's all.
Thank you very much, sir. You managed time to join us. Thank you very much. You inspired us a lot. Thank you, sir. Thank you. Now we are coming to the main course of today's webinar. Nowadays, we all know that modern research has reached such a height that we always remain surrounded by sophisticated innovations. With their huge benefits to ease our life, these innovations impart adverse effects as well. Today, we are going to listen to an interesting talk on how one of the most important discoveries of all time, that is light, can bring darkness to our lives. Moreover, we will experience in the second half another talk on how interdisciplinary tools can be employed in ecological research frontier, those which are not familiar at all, like GIS, remote sensing, telemetry, etc. But still, how they help to overpower the challenges in diverse fields of ecological research. We are fortunate enough to have two eminent scientists as our resource persons to enlighten us in the diverse domains of the biological sciences. Our first resource person is Dr. Oshomanju Chottoraj. Welcome, sir. Dr. Chottoraj received his PhD from Vishwabharati Shantiniketan. In his doctoral research, he added a new dimension to the meiotic cell cycle of oocytes, where melatonin modulates the action of maturation-inducing hormone. The novelty of his science is first demonstration of melatonin receptors in any fish gonad. Starting from his doctoral study, he engaged to unravel the mechanism of chronobiological signal through melatonin and body response. Uh, in his postdoctoral studies in Michigan University, USA, Dr. Chottoraj emphasized on the molecular regulation of production of indole amines in mammals. Finally, he became able to set up Octodon dagus as a diurnal animal model for biological studies. After coming back from USA, he joined as assistant professor in Guru Ghasidas Vishwavidyalaya, Bilaspur. After that, he joined as scientist D in the Institute of Bioresources and Sustainable Development, a DBT institute in Imphal, Manipur, where his research group first time showed the effect of artificial light at night on central and peripheral regulations. Presently, he is in Kazi Nazrul University as associate professor and HOD in animal science department. He has produced 3 PhD students, more than 32 publications, and he completed several research grants. He has received several awards for his research excellence. Now, I invite Dr. Chottoraj to please deliver your speech. Over to you, sir.
Thank you, Dr. Shahana. It's a good opportunity for me to give a lecture on this platform. And I also really thankful to Dr. Halaluddin, Principal, MM College, and for his kind presence. So as Dr. Shahana has told that our group is involved in a research which is really very few groups overall the globe is doing in this particular work. And as today's webinar topic is very much interesting, and here also we are finding a word light is there. So understanding life in light of modern research. So, so I should not waste much time on introduction. I should directly go to the slides because it will take some time to. At that time, I will elaborate what I mean to say. So I am directly going to the slides so that I can start the thing. Is it visible? Is
Yes,
the
yes,
slide
Dr. Chottoraj.
slide
Yes,
is
yes.
visible? Okay. Okay. So today's topic is the understanding life in light of modern research. Now the question is that uh, why the light is important, what life is, and what is the present scenario of today's world. So if I ask, what is the can you define life? If I ask anyone, can you define life? I think till date the definition of life is not been properly told, because life cannot be defined. Then biologically means, what do we mean by life? Biologically, life is nothing but it has some properties. And the most important property and the most significant property of life is the response. Then comes, when you are responding, you need to respire. Respire includes everything. Respire includes the food intake, respire includes the digestion of food, respire includes the processing of the digested food material into forming to energy, which we know that one molecule of glucose gives rise to a certain amount of ATP, which ultimately gives the energy to either any kind of living being. Then when you are respiring, then you will obviously grow. So growth is one of the important property of life. So when you are growing, then that means you are accumulating some metabolic waste inside the living organism. Then that metabolic waste should be removed. Then the process comes the excretion. So first the response, then the respiration, then the growth, then the excretion. And when you are excreting and simultaneously you also need a movement. Now people will say, then what is happening to plant, because plant cannot move from one place to another place? Obviously plant cannot move from one place to another place, but it has movement. Internal movement is there. The movement, we know, when the photosynthesis happens, when the transpiration happens, there is a movement inside the nucleus. So we all this, so movement is one of the important property of life. And above all, but not the least, is the reproduction, because you have to reproduce to give your origin and to sustain your origin. That means this response, respiration, growth, excretion, movement, and reproduction, these are the main property of life. And now the question comes that why the life is present in earth. There are several theories, several models, several hypotheses are there. But for me, what me and other chronobiologists, they have found that the Earth is positioned in an area in this solar system where we have a daily rhythm of 12 hour of light and 12 hour of dark. So we have a daily rhythm and we have an annual rhythm. We have a 365 days rhythm and we have a 24 hours rhythm, right? So 24 hours, almost 24 hours takes for the earth to rotate in its own axis, and 365 days is taking to rotate around the sun, that we know from our primary education. So this 12, almost 12-hour light and 12-hour dark, you cannot find in any other planet, in other 8 planets which we know that, which is the member of the solar system, that they do not have this kind of periodicity. That means this 12-hour light and 12-hour dark is a very important factor for our presence. And if you see that in our higher secondary education, we know that photosynthesis is important and the only process of productivity. So the primary producers are the plant, and ultimately whatever you are taking as a food material, it is from the plant. That means it is the C6H12O6. That means the glucose. And if you see that photosynthesis is having two phases, one is a light phase, another is a dark phase. Then why the dark phase was needed? Was there any relevance of needing of the dark phase? And we also know that ultimately production of glucose happens in the dark phase, not in the light phase. That means the natural darkness is a mandatory thing for the existence and sustainability of life. Now the question comes, which is a actually this question for a prolonged time I was in Shantiniketan Vishwabharati. Though I was studying science, but if anyone visited Shantiniketan and stays for a longer period, till date I can tell you that the seasonality, the effect of seasonality, the color of seasonality, the smell of seasonality, everything till date it can be found in Vishwabharati. And the question came from various writings of Tagore, that what is nature? If I ask what is nature, everyone will answer, I know, the same thing, that the tree, that the the hill, the flower, the river, the ocean, everything includes nature. But except me, we do not consider us as a part of nature. And here lies the all problem, that we never consider ourselves as a part of nature, and we are also a very important parameter or important component of this nature that we do not think. And that is why the main problem comes. So initially, I was, as Atrey told very nicely, that I was in Imphal, because in Guru Ghashidas University I worked for a very small one year time, because I have a tendency to work in Northeast, because that has a specific reason. The reason is, I asked the question that, as we know that in all over the world there are few biodiversity hotspot zone. So what is the hotspot zone? We know that where is the abundance of the animal and plant and microorganisms are more, and which is and according to the presence of this of that organisms, we have divided the hotspot area. And one of the major hotspot we have in our India is the Indo-Burma biodiversity hotspot area. So my question was that, why this Indo-Burma biodiversity hotspot zone is present in that area? Why it is not present in Tamil Nadu? Why it is not present in West Bengal? Why it is not present in Jharkhand? That means there are some parameters which you all know, being a part of the ecological study, that biotic and abiotic factors are very much important for the sustainability and the origin of life. So it has been, so that was my question, because my training in my PhD and postdoc was on the biological rhythm. And then I started to find the question that, why this biodiversity hotspot zone is very much specified to a specific area in all over the world. And that was my first question. Then I entered into the IBSD and started work. And after that, in 2018 November, I moved to this Kazi Nazrul University. In the bottom, in the right hand side, it is the gate of our university. So now, if I go to the origin of universe and development of biological clock. So as we know that the Big Bang theory is the most potent theory now till date about the expansion, or rather the divergence of the origin of this universe, or rather the I should not say the universe. I should restrict to a solar world, and obviously other solar worlds also are facing the same thing. There is a big blast, then there is a scattering of big molecule, then the big molecule also attracted the small. It's just like a tubri. We often use in our pataka and all this thing. Tubri, if you see that when you are giving the fire, it is busting, then it is stopping. But in case of in case of life, what is in case of Earth or other planets, what is happening? It is moving towards around the sun, and actually they have their own axis, and in their own axis they are rotating to their own axis, and also they are moving towards the sun. But the most important thing you see, that all these 9 planets are rotating around the sun, but no one is interfering, no one. If there is a single interference, if the axis or the pathway of their surrounding of sun comes to closer of any planet to another planet, there will be a devastation. And Tagore has told, I know everyone knows the Bengali. So there is a famous song of Tagore during Bashant Utsav we used to sing that Jabar age rangi diye jao. And in that song, Tagore is telling that where that color should go, the color here the color he defined as a life, as a rhythm. And he is telling that Bishwa nacher kendre jethai chondo jage. So it is the center of the dance of the universe where the rhythm should be there. That means rhythmicity is equivalent to a life. If there is arrhythmic to anything, it is deviating the properties of life. That means what is a rhythm? A particular signal in a particular time leads to a particular work. It is relevant to everything. Either it is a physical thing, or it's a chemical thing, or it's a mathematical thing, or it's a biological thing. So it is a mathematics. Basically, everything is a mathematics. So the rhythm, if the rhythm is destroyed, it is destroyed everything. Now the question is that in life, how this rhythmicity is maintained. That means now it is clear that the rhythm means a response of two things. First response is that individual and the surrounding environment, and the individual within the individuals. That means if I am getting a response that outside is hot or cold or whatever it is, that I will receive, and accordingly my internal physiology will go on. That means a biotic and abiotic response ultimately leads to a rhythm. And what are the parameters which these rhythmic things are being sensed? From the cyanobacteria, as we know, that a very primitive kind of origin of life, like virus and cyanobacteria and all these things, and it has been found that in cyanobacteria also it has a clock mechanism. Like we have identified the clock, we know what is the function of the clock, and is the same thing. It has been found in the very various primitive organism, and nowadays it has been found in every organism, every cell. There is a clock mechanism, or rather the clock associated genes. And the most important clock associated gene is the clock, which is the circadian locomotory output caput, and another is the brain and muscle arylaldehyde. And there is a cry, and a cry is basically a cryptochrome. As we know, the cryptochrome is a very much important factor for transfer of energy in any system. And the power or the period. There are other genes are also there, but this clock, BML, and power and cry is present in every organism. In every organism, you will find these four genes at least, and these four genes are very much coordinatedly, they work to keep our biological clock. Now, what was the concept that how this clock, as I told that the time clue or the particular signal in a particular time, if I consider, then initially it was told that it is like a master slave model. Master slave means the brain is having the clock, and this brain is controlling all the peripheral organs. Okay, so this is called a central organ, and other like heart, liver, muscle, kidney, whatever the peripheral organs are there, it is being controlled by the brain, and brain is giving the signal. Whatever the signal, either it is an activity, or it is light is there or not, or you need the feeding time, or you need to food. So everything is governed by the brain, the master. After that, it was found that no, this is not the case. It should work in a matter of orchestration. Orchestration means what? we have seen the orchestra, right? Orchestra means there is a single person who has a stick in his or her hand, and suppose there are groups of instrumentation. One is sitar, one is violin, there is flute, and suppose according to the signal, that particular person is telling to sitar to start the orchestration. Then sitar starting the orchestration, then suddenly he told, okay, sitar you stop and violin you start. And as we know, in presence of music, that violin cannot start from the beginning. Violin has to start where the sitar is ending. That means each and every group of instrumentation needs to follow each and every one rhythm, otherwise it will be arrhythmic. Similarly, scientists propose that no, it should be an orchestrated model. That means obviously brain is having the clock, and likewise every organ like heart, lungs, the heart, the liver, the kidney, the intestine, everyone is having a definitive clock, and this clock also can interfere with the clock of the brain. That means it is a coordination system. So if, and this is a very much scientific thing, that this coordination is very much needed to run a system smoothly. And from then this orchestrated phenomenon started to existence. Now the question is that okay, we have understood that clock is there. There is a cycle of light and dark, there is a cycle of rain and non-rain, so environmental factor and also the biotic factors are also there. Now the question is that who controls, or is there any molecule, is there any chemical substance which can regulate this biological rhythm? It has been found, yes, in case of higher vertebrates, we have a organ called the pineal gland or the pineal gland, which is located in the in front of the third ventricle of the brain, and it is very closely to the pituitary and the hypothalamus. That means, and there is another organ called the suprachiasmatic nucleus. So this particular pineal gland or the pineal gland is photosensitive. But in case of higher vertebrates, this the photic sensation of pineal gland comes through the retina or the eye. That's why this optic chiasma, if you see, the optic chiasma is very much related to the suprachiasmatic nucleus, and which is closely in close proximity of the pineal gland. That means if the environment says that light is there, the pineal gland functioning accordingly it controls. And pineal gland secrete a specific hormone called the melatonin, not melanin. Melanin is different. It's a melatonin. It's an indolamine. You know the amino acid tryptophan. So it is a derivative of the tryptophan amino acid. It's a very small molecule, around 210 Dalton only, and it is conserved from cyanobacteria to human being. So you can understand that how important this molecule is. So this melatonin molecule is mainly secreted from the pineal gland, but it is light sensitive. That means when there is light, it will not synthesize the melatonin. So only when there is a dark, then melatonin will synthesize, irrespective of the habitat of irrespective of the habit of the animal. Melatonin always synthesizes in the dark condition. Like we are diurnal animal, we sleep at dark, and we are active during daytime. And there are animals who are nocturnal, who are active during night but take rest during day. Like we see that the dog, that bat, and all these animals are the nocturnal animals. But the synthesis of melatonin always happen when there is darkness. And now the question comes, then what happens to the blind people then? Because blind people, there is no sign of sending light to the brain, as we know that here pineal gland, if it is light sensitive, then what will happen to blind person? It is found that the suprachiasmatic nucleus has a sensitivity towards our skin, because we know our skin is very much light sensitive, and it has a tendency to understand the rhythm, and the clock associated genes also help to determine that weather, and they are more sensitive for the blind people. And same thing happens to the deep dermal fishes. That means those fish who are residing very deep in the ocean, where light does not penetrate, but they also have a sense of the light and dark. Now this pineal gland, in case of fish and reptiles, this is in case of fish, it has three parts: the end vesicle part, the pineal stalk, and ultimately dorsal sac. And this part is light sensitive. That means in lower vertebrate, this pineal gland is directly, it can receive the light, and it has the photoreceptor molecule, and it can synthesize melatonin also. That means there is a evolution happens from the fish to higher vertebrates, and ultimately the melatonin synthesis, but the synthesis of melatonin, the process of synthesis of melatonin does not broke. So how the process of synthesis of melatonin happens? It happens inside the pineal gland, and it is an enzymatic process from tryptophan. Then the tryptophan hydroxylase enzyme works. Then there is a decarboxylation. Then there is an enzyme, the N-acetyl transferase. This N-acetyl transferase enzyme. So from the then this is the serotonin which is produced. This serotonin we also know is a neuromodulator, and when this serotonin is produced, then there is an enzyme called the N-acetyl transferase enzyme, which ultimately gives the signal whether ultimately melatonin will produce or not. And that depends on the superior cervical ganglia, where some norepinephrine is there, and there is two receptors, beta adrenergic and alpha adrenergic. When dark is there, then beta adrenergic is activated, and ultimately it produces the cyclic AMP, and cyclic AMP triggers the NAD, and ultimately it produces the melatonin. That means this is, we know that that this process is also conserved. This is a very important thing, that this four-step enzymatic process is conserved for the melatonin process, production, starting from cyanobacteria to human being, which is a very important important finding, and it is very much correlated with the process of the evolution. Now the thing is that here we find that light is very much important, because we know that N-acetyl transferase enzyme is light sensitive. So if there is light, this enzyme will not be activated, and ultimately it will not go away to produce the melatonin. That means now the importance of light we have understood here, that if light is more, or light is enormous, then the production of melatonin will be hampered. So the same thing happened in 2014. Nobel Prize in Physics was given for the identification of the LED lights, as we know, light emitting diodes. And unfortunately, the LED which we are seeing right now, it is the blue spectrum light. It is not the white light. That's why the Nobel Committee gave the concern, when they gave this prize, they also give concern that until or unless with this blue, red, and green, if we cannot form a white LED, because when you form a white LED, then you can have variety of light emitting spectrum, because we know the light have a spectrum like VIBGYOR, we all know, right, from the violet, indigo, green, yellow, orange, red. So when it is going towards the red, the wavelength of light is more. So the wavelength of light is more means it has a less penetration power to the organism. And we know the ultraviolet ray, the violet ray, it is bad for our health as it has a shorter wavelength. So shorter wavelength means it can penetrate more. And this LED which we are using right now, it is a blue spectrum LED. Its spectrum is around 450 nanometer. That is a very short, and which is very very harmful to our health. Not only our, starting from plant to human being, everyone's health is in hazardous condition. And in 2015, the Nobel Prize went for the mechanistic studies of the DNA repair. You see how things are happening, how things are contradicting. That when LED you have found, then you have found that the Nobel Prize is giving to the, and then in 2017 the Nobel Prize goes to physiology medicine for the discovery of the clock system, or rather the circadian rhythm system. So what is circadian? Circa means around and dian means dial. That means around 24 hours rhythm. So 1 day rhythm is called the circadian rhythm, right? That is the movement of the earth in its own axis. In a The whole, to move in its own axis, earth takes around 24 hours time. So this is a circadian rhythm, and Nobel Prize was given in 2017 only. So it is the first time people started to believe, no, there is something is happening with the biological clock. Earlier, people have started work from 1950 or with this melatonin and chronobiology, but no one, until or unless you are having a Nobel Prize in this particular field. So this is not only the Nobel Prize for these three people, it is the Nobel Prize for all the people who have supported their work eventually, because this work they have done in 70s and they have got the Nobel Prize in 2017. So if we see how the light has been changing from the primitive environment to the present day. So the sun, as we know, the sun is the only source of light, and it has several wavelengths are there, and accordingly it comes to our planet, and we can absorb that. And then we have found the fire. So it is if you see the color, it is an visible and it is infrared. That means its wavelength is much more. That means it has a less penetration power, and it is less harmful. Then we have found that some other kind of light, like candle light, and it also has the yellow red kind of wavelength. Then we have found the yellow light, that the bulb, which is also yellowish in its penetration. Now we have found the LED, and if you see how the spectrum of light, the color is changing from reddish to yellow, then suddenly it is in blue. That means the wavelength become very much less. That means it has more penetration power, and it can give you more harm than the other lights. And if you see that what is happening, this study was done from University of Michigan, that how the artificial light at night, the presence of artificial light at night, if you see from 1998 to 2013, if you see how it is increasing, if you see this light. So this is the area of our India in the eastern and northeastern area. If you see, this was the yellow dot, means the light at night. And if you see how it is increasing within 2013, and now also it is increasing more. That means the light at night is increasing day by day, and we understand this is the evolution, and this is the development. So if you have more light, our concept is that more light means more development, because till date internationally there is no word of light pollution. That means people do not, if I go and say people or the policymakers that light is harming us and you should, People will say, "Are you crazy? Are you mad? You are coming here to say that light is a pollution? What nonsense you are!" This is the situation. People are not understanding what the light is hampering. And from 2015 onwards, people have started to work because how the light can give you the changes, how the wild organism, how the predation is happening, everything is changing. So people are starting to work, and these are the very good worker on this particular field, and they have already told that the artificial light is really uh, hampering everything. And what after all these things happening, what in short, if I say that artificial light at night, in short, if I say, is ALAN, then ALAN, where it can affect it, it first it affect on the environmental filter. Filter means what? That you are destroying the periodicity of light and dark, right? So the filtration is then the niche. We all know what is a niche means in ecology, the area where organization and the group of organization live. And also it can, this niche and this movement and behavior, that means if there are nocturnal animal, they used to prey at night time. If you are giving light, so their movement is hampered. So their movement is, and as we know, movement is one of the important property of light. And ultimately, when movement or behavior is hampered, then the interaction of the species will be hampered, right? Because in a particular time, there are particular animals or plants who are gathering together, and they have a communication. That interaction is involved. So if these four are affecting, then ultimately your community assembly is affecting. So and interaction strength is affecting. So when you are affecting your community assembly and the interaction strength of an ecosystem, then your community structure in its danger. So your community structure is really in a dangerous condition. Now, when your community structure is in dangerous condition, then your traits and interaction network, the animals and their interaction, is in danger. And ultimately, your function, your stability, your sustainability, everything is changing. So this is a huge impact which we do not understand what light is giving to us. I should not say light, the artificial light at night, because the normal darkness is going out of our life. Now, to do all this, to do some work that what light is affecting us, we thought keep the most important organism which we feel, that the fish, because fish is an organism which live underwater. And as we know that water is already a protected shield for the light, because water, air and water surface, that the interface and the light penetration, it has less light penetration happens in the water. And as and we try to see whether the fish is being affected or not. If fish is being affected, then we can say that the effect of beyond fish is more, because they do not have any shield of water. They are more, they are more exposed to light. That's why we try to and we do this on zebrafish, because as we know, the zebrafish is a very important model organism. It has a genome similarity with a human being, and nowadays it is a very good tool for the biomedical research. And we have started work on zebrafish. And as we know that the collection of blood is also very important, because many parameters, say, if we want to see after the effect of light what is happening in blood, so we have standardized a protocol on the collection of blood. And we because zebrafish is a very small fish, around 4.4 gram in height, in weight of an adult fish. So we have tried to, so we found that the collection of blood is very much important for the study. So we did that one, and we tried to understand that how this, as I told in our earlier study, that the BML, clock, power, cry, and melatonin, how these things are being coordinated, and how they are helping each other. So if this is a nucleus, if this is a nucleus of a cell, then you can see, as we know, in the nucleus there are a lot of factors are there which can start any kind of biological phenomenon. So nuclear factors are very much important, which can give you the synthesis of the BMAL, the synthesis of TPH1, which is the first enzyme for the melatonin biosynthesis. And what happens when there is a nuclear factors are triggering those, then the synthesis of this protein happens outside the nucleus, and this BMAL, and this clock, they form a dimer. So they form a dimer, and they come inside the nucleus. And when they come inside the nucleus, what happens? They also triggers other factors like cry, PAR, and melatonin biosynthesizing machinery. And when this proteins are being synthesized, then this PAR and cry is also form another dimer, and it come inside the nucleus and repress or inhibit the process of clock and BMAL. That means this clock and BMAL, these two genes are triggering the process of biological rhythm along with the help of melatonin. And when they have started this signal, then another set of protein, PAR and cry, they come inside the nucleus and stop this thing. That means, why I am showing this, like our finding of this, because when this clock and that means this is a synchronization. That means if your clock and BML come in a particular time, then a particular signal of forming all these things will happen, and then there will be a formation of PAR and cry dimer. Dimer means two proteins are coming close together and they are forming as a one. So this PAR and cry are coming inside the nucleus and inhibit this clock and BML. That means this process is a synchronized process. That means if this PAR and cry could not come inside the nucleus, then, then the process of synthesis of all these factors will go on. That means the rhythmicity will not be followed. And whether this process are stimulated by light, that we wanted to know. And I'm not going to the details of this. I'll go to my cartoon slide. So now, as we know that that reproduction, excretion, movement, feeding, all these are the peripheral things, right? This is not a central thing. So now the problem is that if the central system and peripheral system are coordinated by each other, then whether this light can give a factor to the reproduction. Why we have chosen reproduction? Because we know the reproduction is one of the important parameters for the sustainability of any organism, and it is being induced, whether it is being induced by light or not. So what we try to see there. So so this is we kept fishes in two conditions. One was continuous light condition and one was in the continuous dark condition. So as we have told that light means less melatonin and dark means normal or more melatonin, right? So in comparison to the light, when there is light is there, melatonin is less. When there is dark, melatonin is more. So it is happening in the brain. Then what is happening? So as I showed in the structure that thalamus and hypothalamus is very much close to the pineal gland, then in the here we know that pituitary is the master gland, but it has also some neuropeptides which can also induce the synthesis and work of the pituitary gland, right? So in case of reproduction, there are two kinds of neuropeptides. One is gonadotropin inhibitory hormone, and another is gonadotropin releasing hormone. And remember one thing, in course of evolution, the negative factor is the factor for the evolution, not the positive factor. So what is happening when melatonin is less? Melatonin is less. What you are seeing? Gonadotropin inhibitory hormone is high. Gonadotropin inhibitory hormone is high. And when melatonin is more, gonadotropin inhibitory hormone is less. Right? But in both the cases, whether melatonin presence is more or less, gonadotropin releasing hormone is always high. That means the positive regulator is always positive, but the negative regulator is negative when melatonin is uh, more. That means it is high. High means it will not process the reproductive phenomenon. Now, what this, if melatonin is less, what this GnRH is doing, because as I say GnRH is more at that time, then it is inhibiting the luteinizing hormone and follicular stimulating hormone to synthesize from the pituitary. This luteinizing hormone and follicular stimulating hormone, what it does, it goes directly to the gonad. Here it is the ovary. And you see, when it is going directly to the, so it is less in amount, and when it is less in amount, then other factors, other growth factors are also high, but the maturation, mainly the cyclin and CDC, these two protein needs to be united, and this is called the maturation promoting factor. So this is basically a progesterone induced phenomenon. So when this cyclin and CDC is not bind together, then the ovary cannot be fertilized, the ovary cannot be matured. But you see, when melatonin is in adequate amount, there is an inhibition in the GnRH. GnRH is always high. So when GnRH is high, the luteinizing hormone and follicular stimulating is also high. And ultimately, this luteinizing hormone and follicular stimulating hormone forms this cyclin and CDC as a maturation promoting factor, and it leads to the maturation of the gonads. And here you see, when there is melatonin more, in ovary also, the melatonin is also more. But when melatonin is less, here in ovary also melatonin is less. That means what we have understood from here, that if you are exposing animals to light, it gives a signal through the hypothalamus, pituitary, and gonad axis. So here it is hypothalamus, then it is a pituitary, then it is a gonad. So this is directly being influenced by the melatonin. Now the question is, okay, we have understood, so melatonin. So what we have understood, that if there is more light, then the maturation of the gonad is being inhibited. And this kind of phenomenon is very much randomized nowadays in case of human population, that polycystic ovarian syndrome. What is PCOD? PCOD is nothing but that your ovary is being matured, but ovulation is not being happening. So every month there is an engagement of more ova, and ultimately you have a polycyst like phenomenon. And I am not telling that light is the only cause, but light is one of the major cause of happening, and people have already proved this. I am not going to that particular phenomenon, but after our this work, people have accepted this thing, that yes, light may also have a very important phenomenon in inducing the polycystic ovarian syndrome. And melatonin is being used nowadays as a clinical trial for the reduction of polycysts in case of ovary. So this is a very much important phenomenon, that when there is light, melatonin is less, and population in the ovary is being very much, the ovulation is less. Now so this has been published in I think in 2017 we published this. Uh, then we wanted to know that uh, yeah, molecular reproduction developed. Yeah. Now we wanted to know what. So this was a very short-term method. We have given this thing in the that animal were exposed for around 5 days. Now we wanted to see what happens if we can expose for a prolonged period, and then we started the work for 1 year. And you see, this is, and then we tried to see this phenomenon in case of clock associated gene. So this is a normal light and dark situation. You see that, as I was telling, that clock and BMAL will form a dimer. You see the peak time of synthesis of BMAL and peak time of synthesis of clock is similar. That means they can form the dimer, and the peak time of synthesis of cry and peak time of synthesis of PAR are similar. So they can also form the dimer. So this, but when you're exposing them to light, you see the thing. There is no parity. It is giving peak here, it is giving peak here. This has no rhythm. This has a very insignificant rhythm. That means the whole rhythmicity and BML clock PAR cry is being lost. What is the factor only? It was only light. Then we have seen the level of melatonin in serum, in brain, in retina, in ovary, everywhere. If you see, the level of melatonin has been reduced. Then estrogen level. Estrogen, we know that estrogen is very much important for the growth of the gonad, right? The growth, that is the structure of the gonad is maintained by the estrogen. So what happens? Your ovary is being producing normally. Ovary is healthy, no problem. But your progesterone level is low due to the light. So when your progesterone level is low, estrogen level is continuously high, then ovulation is not being happening. So it is the reverse phenomenon of what? It is the reverse phenomenon of the natural pills, that the pills for the birth control we take, where progesterone level is high, here progesterone level is low. That means your ovary is ready to ovulate, but the ovulation process is being delayed. That is, the maturation is being delayed. And the TNF alpha and NF kappa, I am not going to details because it might be, because these are the inflammatory things, and we have found that in ovary these things are also being increasing. And simple eosine hematoxylin study also sees that there is a tumor like growth happens in case of zebrafish. And we have studied the transcriptome analyzing, and we have found that with this light exposure, the most important signal which is being happening, affecting, is the environmental information process. That means continuous light exposure is hampering your environmental information process. You are being less responsive to the environmental clue. That means, and other things are there, but the most important thing is the environmental clue, which is being affected through this light exposure. And what we have found, that when you are exposing the light continuously, this clock BML synchronization is lost, PAR cry synchronization is lost, melatonin is less, the tumor inducing genes are more, NF kappa beta is more, TNF alpha is more, and it is ultimately giving you the phenomenon of a tumorigenesis. Obviously, it is a benign kind of tumor, but the reproduction ability is being lost. This ovary cannot be reproduced, cannot happen. So we have published this in 18 in Science of the Total Environment. And after this publication from the France, they have a group, because they have started to work on this artificial light at night. And they have formed this is the first ever made any committee in this art, that against the light, against the use of light. And they were very happy that from whole this Southeast Asia, there is not a single work, then only our work they have found. And they were very happy to incorporate that in their report. And also we have tried to write a perspective, that as we know, that in case of asylum, in case of cell, I mean, in the prison, the light is whatever the light is present, it is in very close proximity, or it is very less. So the condition of light dark cycle in case of prison is very tough. So as we do not have the ability right now to work on this, so we have written a perspective in chronobiology in medicine. And they have accepted our thought. And one group in Paris and one group in Netherlands, they have started to work on this particular thing, that how this can be useful for the prisoner, and what kind of phenomenon they are happening. And now I come to the effect of coronavirus, like COVID-19. So recently it is accepted in 29th of July. So we have wrote a review, and we have very systematically did one thing, that why this kind of pandemic, or the EIDs, that we call that, the infectious disease, emerging infectious disease, why these infectious diseases are more. If you see that coronaviruses are a group of virus which has a normal abode of bat. So bat is their main reservoir. And starting from the Spanish flu to modern coronavirus, in this 130 years old, there is an increase in the light population. And we have tried to give the evidence that how this light population is being increased, and how the bat population is being decreased. And when the bat population is decreased, and bat is not a wild animal, bat is a very much domesticated animal. It generally, you can find bat in our locality. But last 25 years, 80% of the bat population has been endangered, and now the status of bat is in endangered category. So if the viruses which are normally an abode to this bat, if bat is being removing, virus has to survive. So how they are going to survive? They are trying to find another host. And in that case, human being is the most prevalent organism which is present in front of bat. Because if you see, then these viruses try to mimic. First happened in case of the SARS, Southeast Asia respiratory syndrome. Then happens with the MERS, Middle East Asia respiratory syndrome. Then ultimately this COVID-19. So they are changing their pattern for their survivability. And who is responsible for this? We. Because we are destroying the trees, we are destroying the normal habitats of animals, and we are giving light. And this light is the LED light. And it has been also reported the photosynthesis is also being hampered due to the presence of this artificial light. So in this review, we have tried to emphasize that how this artificial light at night is related to this COVID-19 outbreak. Obviously, this is not the whole reason for this outbreak, but this is an important factor, that if your reservoir is being destroyed, then the organism residing within that reservoir, where they will go. So they have to find somewhere else, and we do not know what is happening to cows, what is happening to dogs, what is happening to cats, because they are the very much domesticated animals of our life. And the cats and dogs, they are the nocturnal animals, and we are using light like anything. And for our thing, every time we are with the mobile, and nowadays online thing, virtual thing, we are very much proud that, oh, we are doing all these things, we are so developed. Wait, what? Because we are desynchronizing everything. What we are desynchronizing? You see, our sleep, our muscle reaction, our pancreas ratio, our cardiovascular system, our everything. If you see where we are, even we have found that the increase in the, oh, what is diabetes? If I say what is diabetes, what is cancer? It is nothing but a loss of signal. For diabetes, either you have less amount of insulin, or you have a bad thing in your reception. So either your receptor is wrong or your insulin is wrong. That means the synchronization between these two are lost. What is happening to cancer? Cancer is nothing but when there is we know that every cell in our body except the nerve cell has a definite period of time to, cells to go to their cell cycle, and that period of cell cycle is being arrested somewhere. So if this arrest is being withdrawn, then there is a continuous cell division happens. So if it happens to any organism anywhere in our body, then there will be a formation of tumor. And if this tumor is being again more, and it is spreading more, then it is a malignant. So basically, whatever the lifestyle disease we are talking nowadays, starting from the blood pressure to cancer, all are a process of desynchronization. And light is playing one of the major factor for this. If you see this slide I made, where we tried to see that from agriculture to human health, everything is being affected by light. The prey predation system is being happening. It is being destroyed. The agricultural thing has been destroyed, because the relationship, the light, presence of light and absence of light, basically we are adapted accordingly. Our system is being adapted accordingly, and we have totally destroying that thing. Many big people will say, you see, this is our system, we will adapt for everything. But are we? We are not able to adapt, because we cannot play with light, because light is the primitive requirement for the existence of life in this universe. So this is our group. So this is Jishan. He has completed his PhD, and he is now in South Korea doing his postdoc. He is Thangal. He is doing postdoc in Wayne State University. He is Rajiv. He is a DBT RA in ICR Institute. This is Sanjita, is assistant professor in the Central Agriculture University. This is Jyoti, who is about to submit her thesis. And this is Gopinath, he is also about to submit her thesis. And this is Rahul, who is always helpful for us in maintaining the fish and everything. And this is the funding acknowledgement: the Institute of Bioresource and Sustainable Development, Department of Biotechnology, Science and Technology, CSIR. And now I have a question over here. I mean, I do not know, because as it is a live, every time when I give a lecture, I ask this question, whether the sun is setting or sun is rising. So it is very tough to say from this picture that whether the sun is setting, and even in normal condition also, if you are there in the morning or in the dusk, it is very tough to say whether sun is setting or rising. And this is the way we are thinking, we are developing, but is it really we are developing? I don't know. Thank you very much.
Thanks a lot, Dr. Chattoraj. Such an illuminating speech, knowledgeable speech you delivered. Really, we couldn't have imagined that artificial light can bring such harmful effects in our lives. Really, it is very
Yeah,
knowledgeable session.
yeah,
Thanks
yeah,
a lot. Now this session is open for questionnaires. Actually, you have make everyone understand such nicely that a few question arises among the listeners. I
yeah,
can find here. This is very nice
yeah.
session, and many listeners have admires about your session and about your understanding, making us understanding the process. Okay, a few questions are there. Uh, Shashwati Sen
Mm-hmm,
has asked
mm-hmm.
that what function occurs in our mind through which we can recognize the color of light. Okay, this is the gist of the question. She asks, what function occurs in our mind through which we can recognize the color
Yeah.
uh, in the
So
white light? Mm,
yeah.
she has asked.
Yeah,
Mm,
so the thing is that as we know that light is mainly received by the retina in case of evolved mammals. We have retina, and retina can receive the light, and that goes through the optic nerve to the brain, the optic center, and basically we can see three colors that we know: that blue, green, and red, which is the basic
great.
color. And then brain has the capacity to mix match those things. So if a person is a color blind, then those processes are hampered, that we know. So basically it is the work of the optic center of the brain, and that optic center actually matches with the color. So as a person, in case of human being, as a person grows, the sensitivity towards the color appears more. So it's a learning process, and ultimately the center in the optic center but basically we see from the nature, we see in our retina receives in blue, green, and red. Then it goes to our optic center, and then ultimately it reflects.
Okay. Thank you, sir. And another, she has asked about what you told about the blind people. Just now we have
Yep.
told about the blind people. She has asked that blind people can't recognize the red and green color.
Yeah.
Why it is specific for red and green color, not for other colors?
No, it is not like that. The blind people, there are two kinds of blind things are there. Either one is your retina, I mean the receiving of photon is damaged. Another thing happens, your receiving of photon is good, but the optic center, the information goes to the optic center through the optic nerve is damaged. Right. So there are two kinds of blindness can happen. So if I consider that both the things are not there, suppose either light is not responsive, so it is light does not only means, it also means a sensation in the form of temperature also, right? And our skin, as I told earlier, our skin is the largest organ in our body, and every organism, skin is a very important factor to recognize the light sensitivity. So it is not the red, blue, or green. It is the
Okay.
matter, and the suprachiasmatic nucleus and superior cervical ganglion. These two are the major area in the brain which can interpret through biological clock system. That means because our blind system can understand this is morning, this is night, we need to see, because this is being entrained. The terminology entrainment in biological clock is very much important. That you are suppose one night you have not slept, so next day early up to 11 o'clock in the morning you slept, or 12 o'clock in the morning you slept, then you will feel that there is some difference in your body. So it takes around 2 to 3 days. Normally, in our familiar language, we say that if you are not sleeping in a single night, it will take around 7 days to mimic that
Exactly.
thing again,
Exactly.
entrain, re-entrain. So that means your clock system needs that much of time to re-entrain your
Okay.
body system.
Okay.
So melatonin also plays a very important role in re-entrainment of the body system.
Thank you. I hope she has understood this. Okay, another question is there. Bhashwati Mukherjee asks, many times the main reason of PCOS and PCOD is said to be anxiety and lifestyle. Is there any cure to this, which can be a permanent cure to these diseases?
My question, I have a counter question to her. Uh, in 40 years ago, 50 years ago, was anybody heard the name of PCOS?
Yeah,
No, no,
exactly.
right? That means these are not a disease. These are all symptoms. These are all arrhythmia. Anxiety stress means what? You have more corticosterone, you have more adrenocorticosterone, then it gives more amount of glucose to your body. So if you have more glucose, then all these are desynchronized state. Yes, you can cure. You can cure by maintaining your lifestyle properly. First
Okay.
thing is that your life. I mean, how you are maintaining, how stress you are getting, how much you are doing with your mobile. Kut, 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 kut. So those things are very much important because nowadays mobile and laptop is just like our heart. We cannot think of anything without the mobile and laptop. So these things, you see, I am giving this lecture with my laptop, and I have around 3 foot gap with my system. But it is nowadays, it is a very much. Now you see, you cannot move outside, you cannot mix with your friends, you cannot go to a party. What you are doing, your movement is restricted, and you are caged with this 4-inch screen, always with the emission of the light, blue light. So what your body will do?
Yes, exactly.
So it is
Serious
curable.
threat indeed.
In your hand. It is in your hand how you will process it with your life. All the lifestyle diseases is curable if you are aware. People are earning money with the diabetes because that, okay, once you are getting a diabetic medicine, these are the bullshit. If you are aware, no, I am not doing physical exercise properly, I am in stress condition, you have to be careful. You have to be very. Now we okay, bhai. Medicine will do its own work. I will do my own work. That cannot happen. You have to synchronize your lifestyle. That's it.
Yes, yes. Thank you, thank you, thank you, Dr. Chattopadhyay. I also hope she has got her answer. Mm, okay, no more question I can find. Uh, okay, I think the questionnaire session is over. I think so. Okay, so thank you very much, Dr. Chattaraj, once again. You have given us such time with us. You have spent and you delivered such a nice lecture. Thank you very much, and we hope in future you also will be with us whenever we call you.
Sure, sure. It's my pleasure, and thanks to you, Atri,
It's our
for
pleasure. It's our pleasure, rather,
no, it's
to
it's
have
a it's
you.
a good, yeah, it's a good thing. Thanks to all for listening and giving me the opportunity. Thanks. So take care, be safe, and less use of LEDs as you can.
And
Okay, bye.
thank you, Dr. Chattar, for your suggestion.
Thank,
We'll try to
thank,
follow this.
sure,
Thank you.
sure. Thanks.
Thank you. Now we are in the beginning of our latter half. Uh, uh, we are beginning in the beginning of we are in the beginning of our second session. Our second resource person is Dr. Shobhuj Bhattacharya. Welcome, sir.
Thank you.
Dr. Bhattacharya has obtained his bachelor's and master's degree in zoology, and then he obtained his PhD in wildlife sciences from the prestigious Wildlife Institute of India and his postdoc from the esteemed Indian Institute of Sciences, Bangalore. Since the last 12 years, he has been actively engaged in research which aims to understand the impact of climate change in species ecology, which lives in the highly seasonal environment such as high altitude Himalaya. Though his majority of works are focused on a small mammal species, Royal Spica, Ocotona royale, in western Himalayas, but he also got the opportunity to work on other species such as American pica, Ocotona princeps in Rocky Mountains, USA, as Fulbright Nehru Fellow at University of Colorado, and on the Eurasian otter Lutra lutra in northern England as British Commonwealth Professional Fellow at University of Sheffield, UK. He has also received training in theoretical ecology, such as evidence-based conservation methods at Bangor University, Wales, UK, as Ravi Shankaran Inlax Foundation Fellow in Conservation Science. Presently, he is working as grant advisor in DBT Wellcome Trust in Hyderabad. Now I invite you, Dr. Bhattacharya, to please deliver your speech. Well, over to you, sir.
Yeah. Can you hear me clearly, Atreyi?
Yeah, I can hear you.
So I have a few disclaimers. So I just today morning, I realized that my neighbors are making some kind of changing in their house. So there will be random noises coming in between. So kindly forgive me if those typical development works related noise comes in. So I just beg your pardon beforehand. Okay. So I'm going to share my screen and then we'll have a very informal sessions where I'll be providing some of the stories about my work and how I use different tools, and some of the tools which I actually did not use, my friends use, but it is a very commonly used tools nowadays in ecological sciences. So I'll be sharing my screen now. Just I probably have to close my presentation.
Yeah.
Just I will try because it's now not showing any application in the window. Just a minute. I have to close probably again.
Okay.
Sorry for So what happens is sometimes these PowerPoints are behaving weird. So we had to close multiple times because I was listening to wonderful lecture by Dr. Chattaraj, and it has been long since my presentation is open. So maybe there's a reason. So.
Okay. You take your time. No issue.
Yeah, I got it. Yeah.
Okay.
Let me know when you're able to see my screen, okay?
Yeah. It is seen.
Can you?
Yeah.
Okay.
Yeah.
So thank you so much, Atreyi and MMM College, to give me opportunity to talk to undergrads, postgrads. I heard that there will be some senior members also who might be listening to this. So this is primarily informative sessions regarding interdisciplinary tools for research in ecological sciences. So I'm primarily now working as a grant advisor at a funding agency called DBT Wellcome, which funds research, primarily biomedical science research in India. But I also worked in Species Survival Commission in Switzerland and Gland, where I actually provide information which is needed for species-related conservation plans, action management plans, which helps us to conserve certain species. For me, it is my animal called Royal Spica. So before starting my presentation, let's have a like a recap of our idea. What is ecology? Ecology is a study of relationship between living organisms, including humans, their physical environment, and it seeks to understand the vital connection between plants, animals, and the world around them. So this is the Ecological Society of America definition, and I primarily believes you can have different kind of ecological definitions, but this summarize what ecology means. So now you have, if you tease apart different components, we are there as a human, as Dr. Chattaraj says, that we often do not consider us to be part of environment, but we are very much part of environment, plus the animal, plus the plants, plus the microorganism. So ecology does not mean only to study animal or only to study plants or only to study microorganism, rather interaction of them in time and space. So this called cloud, a word cloud. So here I try to put some of the words which define my journey as a researcher during my PhD and postdoc in last 10 or 12 years or so. So I primarily consider myself as an ecologist or wildlife biologist. I walked on pikas, which are rabbits. I'm going to talk about more in next few slides. I walked in mountains. In alpine ecosystem, I studied microclimate, I studied population genetics, foraging ecology, behavior, thermal biology, plant-animal interaction. Also, I studied kangaroo rats in California, in Baja California, which is very close to the Mexico border. And we try to look at different kind of people, land use, land cover practices, how that can affect the endangered species kangaroo rat. Also, I studied Eurasian otter, as Atreyu mentioned, in England, where otters' numbers are going down significantly in recent past. So what I studied in England is using genetics as a tool to understand what otters eat. Okay, so and then when we know what they eat, we not only can conserve those habitat, but also can conserve their food item to ensure long-term survivability of those animals. So first tool is known as field ecology, which I believe is what I'm doing for my PhD and postdoc. So pikas are relative of rabbits, which lives in high altitude mountains. For my field ecological research in high altitude Himalaya, I had to found a model system which helps me to understand how climate change or oscillations in climate can affect an animal. So now that is really challenging for India, because a lot of us know that we do not have long-term climate data or long-term ecological data like United States or Western countries like Europe. So we need to have a particular species which can tell us if climate change is happening in few years or last 10 to 20 years, how a particular species gets affected, which might play a very important ecosystem services, such as being a prey species. So prey species means several animals which lives in high altitude zones, such as snow leopards, red fox, jackals, weasel, and martens. Everybody eats on this particular animal as their food. Now, these animals look like a rat, but they are lagomorphs. That means they are related to rabbits. They are tailless rats per se, but they are not rodents. So because they're rabbits, they do not sleep during winter, or they do not hibernate during winter. So they are only important food source during the winter times. So these all animals, it can be snow leopard or other animals I mentioned like red fox, they solely rely on this particular animal for their winter food. Now, these particular animal in United States, where people are studying them for long, are known to be get affected by climate change because they have very thick fur coat to live in high altitude Himalaya, and they get heat stress quickly and die. So if they die, directly that will have an important impact on the ecosystem services they provide as a prey species. That means if there is no food, then even you spend crores of rupees for snow leopard related conservation. If there is no food, you can actually not save snow leopards. Therefore, studying pikas is important, and I selected this species for my field ecological study. So, important fact. So some of you, I'm pretty sure know this, what it is. It is called Pikachu, and Pikachus are like inspired from pikas, which lives in Japan. They are a little different than the species which I studied in Himalaya, because Himalayan pika do not make sharp call. Whereas when I went to Japan, in Hokkaido Island, northernmost part of Japan, close to Russia, I found they are really, really loud, and obviously they are very cute. And obviously, and those cuteness and their availability in the ecosystem, their important role, what they play in the ecosystem, inspire Japanese artists to make Pikachu cartoons. So for my field ecological study, I went to Kedarnath Wildlife Sanctuary. So now Kedarnath Wildlife Sanctuary is in the state of Uttarakhand, which is the northern part of India. And I selected altitudinal range of 2600 meter to 3680 meter in an area called Chopta and Tunganath. So this particular area is really famous for one of the famous shrine for Shiva called Tunganath, which is said to be India's highest Shiva temple. But I know I went to different places in Himalaya, so I have the GPS location. I know that's not right. So there are some Shiva temples in Himalaya which is a little higher than this. But then this area is also famous for beautiful scenic views, as well as plants and animals which lives here. So these are the faunal diversity which you can see in my study area. So this is Himalayan thar. It's kind of ungulate like goats. Himalayan monal, it's a beautiful pheasant. That means it's really big bird. Size will be like can be like a chicken, bigger. Himalayan black bear, and one of the most elusive. That means you have to be really lucky to see a masked deer. Often this masked deer get hunted for their masked gland, but this Kedarnath Wildlife Sanctuary, also known as Kedarnath Masked Deer Sanctuary, because masked deer lives there. These are the predator which eats pikas. Okay, so the common kestrels are the raptors, Himalayan weasel, common leopard, red fox, and yellow-throated marten, and as mentioned before. So if you go to my field in Western Himalaya, primarily in Kedarnath or at around in other areas in Kedarnath, you can see pikas lives in two different kind of habitats. One is human-made structures, because different pika species we can found in Himalaya, like 9 different. Few of them can make burrows, but then lot of them cannot. So my species, Royalist pika, cannot make burrows. So they use human-made structures, such as this wall in the picture, or they can use the empty spaces available between rocks, okay, known as talus. Those natural taluses they use, and the spaces known as crevices. So they use those spaces for their nest or their habitat. So this is one of the pictorial representation of my study area. So my study area have a subalpine region, which is primarily forested between 2600 meter to 30 to 50 meter elevation. And then I have permanent plots, which are 50 by 50 meter plots. Now, how I came up with the idea of 50 by 50 meter plot? 50 by 50 meter plot idea I came up with the idea that my animal, being a tiny small animal which weighs around 100 grams or so, very small, like your fist or like a small hamster, they use in a particular day, their home range size is around 50 by 50 meter. So my plots where I have to study as per my field ecological study, where I have to study their behavior, count their number, what kind of food they eat. So I selected those plots. Total, I took 10 plots so that I have enough replicates from subalpine to timberline and alpine zone. Now, timberline is a very interesting term. So just let me explain it. So I hope you can see the cursor or my mouse moving on the screen. So there's a line kind of area in this picture, above which there is no tree. So that's called alpine region. and this line known as tree line or timber line. So picus lives below those lines called subalpine till the alpine zone. So I studied a variety of different things. So today, because we have very limited amount of time, so I want to give you a flavor of only one of the components where I studied their activity. So I'll be discussing about sampling protocol as a part of field ecological study. One, like how one can go about it. So what I did is I observed their activity period during May to August for 5 years during my PhD. So now why I selected May to August? Now, as I mentioned, they're thick fur-coated animal. They have to live in high altitude, also have to survive winter. They do not hibernate. But then during summer, if it's too hot, they get heat stress very quickly because their critical lethal temperature and their normal body temperature are 3 degrees different. So very quickly they get heat stress and they die. So peak activity period I selected when they have the higher probability of getting heat stress. So I observed them during morning 6 to evening 7 in an hourly interval, such as 6 to 7, and I spent, how many picas I can see, they are active or inactive in my plot or not. And how I calculated that as an activity rate is number of individual observed in my hourly occasion, which is 6 to 7, 7 to 8. And I divide those number with the maximum number of individual I observed during the day. For example, between 6 to 7, I have observed only one animal. But during the entire day, which is 6 to evening 7, I have observed probably 10. So my activity rate for morning 6 will be 1 divided by 10. And then I plotted that to measure activity, okay? I also collected microclimatic data because I try to look at what is the adaptation strategy these animals actually opt for to avoid heat stress. So what I did is I used a small tiny eye button. It's like a 5 rupee coin in the topmost picture. So they cost like around $25 or so. And you have a launch pad and you connect that to a computer and you can define you want these eye button to take temperature data, and sometimes if it's a very expensive one, it can also give you moisture data and some other lot of different kind of data. Because I had a very tight budget with my PhD, I could only afford to have a data eye button which gives me temperature data. So now temperature I have, and then those temperatures are collected in every 10-minute intervals, and I club them together, make an average temperature for an hour, like 6 to 7, then average temperature for 7 to 8, and I collected a moisture of the soil using hygrometer, which is the second picture. And sometimes it's difficult to get soil temperature, so I use a soil thermometer using as the last instrument in the picture. These are different behavioral states. So I studied these behavioral states for a different purpose, different altogether, but I thought it's good to give you some kind of idea. So they can be found in feeding, they can be also seen running, they can be also seen having musing. That means this animal just sit like a monk doing meditation in a small, beneath a small rock. The reason for musing behavior are not known, but they believe that it happens early morning and late evening hours. So they are said to actually help the animal to acclimatize with the climate as well as look out for predators. They also groom themselves, the licking and cleaning themselves from the parasites. Also, they have to be very vigilant because I already showed you there are a lot of predators abundant in their habitat who can actually kill them. So they have to be very vigilant to see who are there before they venture out from their crevices and start eating plants. So here is one of the examples of my field ecological study where I try to understand influence of microclimate on pike activity. So these 3 graphs, the lower below graph belongs to the subalpine zone, which is the lowest elevation zone. Middle one is the timberline zone or tree line zone, and the upper one is belong to the alpine zone. The dark blue color is the air temperature, light blue color is the burrow temperature or the crevices temperature, and the green color line indicates soil temperature. And if you look at the x-axis, which is the below, those are the different time of the day, okay, 6 to 7, 7 to 8, so on, till 7 p.m. in the evening. So just looking at it, you can see early morning and late evening hour, that is 6 o'clock, 7 o'clock in the morning or 6 or 7 in the evening, bar temperature and air temperature is primarily quite similar. Whereas during the midday hours, where it is around 11 to 2, the difference between burrow or the crevices temperature and air temperature become higher. Now plot the activity which I already told you that I collected and what kind of pattern I got. So the activity showed a very interesting pattern. So they are primarily follow this temperature gradient. That means they are really active early in the morning and late in the evening. Whereas during the daytime, they have an adaptation. So they use this talus where they live or this burrow as a thermal refuge. That means they shelter inside those burrows or the talus to avoid really heat outside. And then they come out in the evening or like later hours, like 3 p.m. and onwards, to avoid, just to eat and get energy. Now, if you compare these graphs, you can see in the alpine zone graphs, which is the topmost graph, they are much more active during even midday hours than the lowest graph, which is the subalpine graph. The reason being the amount of temperature in the lower elevation is much more higher, and it remains high for the longer duration of the time. Whereas in the alpine zone, it's in the higher elevation. The air temperature reaches to a peak, but then goes down quickly. So the amount of activity is also comparatively higher in alpine than subalpine. But what does it mean if when you think about how climate change can affect these animals? So I also did some statistical modeling, which I'm not presenting here today. But then this is a cartoon representation about what does it mean in a changing environment. So when temperatures goes up, my results indicate their activities goes down. So picas lives in the lower elevation zone will show low activity rate during the midday hours, whereas picas in the higher elevation zone will be comparatively more active. Now you have to remember these are the herbivore animals which does not hibernate during winter. So they always have to eat during the growing period, that is when there are a lot of plants available, because during winter the entire area is snow covered, only lichens and few other food species available. So high altitude animals, where a high altitude region where these animals are active higher state, so they will not face much problem if climate change happens and temperature become higher, because elevation will nullify some of the impact of the higher temperature. Whereas in the lower elevation areas, these animals which might face a lot of restriction to go out during the midday hours because they will get heat stress. That means they have less opportunity to get food, which will reflect to their nutritional efficiency. And we all know if you do not have much food in your body, it will definitely going to affect your other processes such as reproduction, ability to survive in harsh conditions, ability to defend different diseases, and etc. The second tool I'd like to talk about is the conservation and population genetics. So here people use various concepts of genetics to understand mating strategy and dispersal pattern in animals, to understand gene flow among populations, like how different genes or the information of the genetic material moves between one population to the other, one individual to the other individual. Also to estimate genetic drift and inbreeding. So I use these for my postdoc. So I went to 5 different areas in Uttarakhand, unlike my PhD where my entire study area is only focused in Kedarnath Wildlife Sanctuary. Now I went to Harki Dun, Madhmaheshwar, Tunganath, obviously I studied there for long, Rudranath and Bedniyal. So they are distributed in like very large landscape, geographical space, and with a various different kind of elevation zone. And each of those places, what I collected is the fecal sample. The fecal sample actually very important to like tools or important material to collect DNA, because any animal's feces or the fecal sample have a layer of epithelial cell around them. Each of the epithelial cell also have DNA of the host. For the pikas, fecal sample will definitely have pica DNA, as well as what kind of plants they eat, some kind of microbacteria, but definitely I can get some DNA out of it. And this is useful because while collecting this fecal sample, I do not have to bother or torture the animal or have to capture the animal. Therefore, without disturbing the animal, without disturbing the habitat, I can actually collect DNA. This kind of techniques called as non-invasive techniques of collecting DNA. After collecting DNA, there are several processes to extract DNA from those fecal samples. And what I amplified or looked for is a microsatellite, which are short sequence repeats, which helped me to identify different alleles. So I know all undergrads and postgrads know what alleles are. So these each colored pics tells me for a particular alleles if a individual is a heterozygous or a homozygous. So now when I know this information, it all get together in a form of a tabulated sheet where you have a lot of numbers and you use statistics, some extent of mathematics and probability to find out genetic diversity, population structure, genetic bottleneck, and also evidence of inbreeding. So here is my results. So the top graph indicates each of these small circles, different colored, indicates an individual which I sampled during my postdoc, and also the color also depicts which area I have found. So you can see there's one of the circle, two of the circle, there are a lot of intermix, and there is a black dot, there's a green dot, there's a pink dot, all together. This means here these individuals actually meeting with each other, and there is a lot of gene flow among them, whereas you can see there is a blue dot which is primarily isolated, whereas red dots are also isolated. And when I looked and mapped them in a Google Earth map, I found the reason. Why the reason is where there's a mixing. Now look at the cursor or my mouse, Madhumaheshwar, Rudranath and Tunganath. These three places are connected, or they all also are in the same mountain zone. So there is no rivers, no lowland valley which separate them. So actually pikas probably can disperse or move between, move between different places. And between Madhumaheshwar to Rudranath, Rudranath to Tunganath, and so their genetic information gets all mixed up. Whereas Harketu, which is red dots, is separated by Mandakini river and lowland valleys, whereas Nanda Devi is separated by Alaknanda river and lowland valleys. So these rivers we found as a semi-permeable barrier for gene flow, because pikas as a small mammal cannot cross those rivers. Second tool I'd like, third tool I'd like to talk about is geographical information system and remote sensing. So we all know there are a lot of satellites which actually always moving around Earth and taking images and giving us a lot of information. So when sunlight hit each object on Earth's surface, and they reflect solar radiation. So they have a different reflectance for forest, different for water, different for grass, different for built up areas such as road or houses. And these satellites actually take those images. And how those images look like? So each of those images for land cover, for water quality, for topography such as elevation, which slopes, north facing slope or south facing slopes, all comes as a satellite image. And people use those images to make distribution model for animals to find out change in the habitat. For example, the satellite, based on satellite image you found, there are 1 square kilometer area available for a pond in 2010, And if you have multiple years of the same data, by 2020, you can actually calculate based on each year's satellite data, how much amount of area of such ponds goes down or up. And that can be habitat for fish, that can be habitat for rotifers and a lot of other organisms. They also calculated the area cover. That means how much is the area is occupied by a big banyan tree versus agricultural field versus say a typical forest made of conifer trees or something. So depending on a research question, people use the satellite images. So I use the satellite image to understand climate change because satellite images and the recent advancement in statistics and several software can allow me to actually predict what might happen in 2050 and 70. So these are called habitat suitability models. So I use 2010 data and versus I try to use different simulation computation model to find out what will happen in future. So for the local distribution data, I have to have GPS locations. So I went to different areas in Kedarnath Wildlife Sanctuary, Kachulakhara, Madhmaheshwar, and Rudranath. Whereas I also collected data from Nanda Devi Biosphere Reserve. It's a really large area. So I get to go to Valley of Flowers National Park, which is here the picture you can see. Nanda Devi National Park also some areas is not allowed to go. So I collected outside of it from Latakhara. And I also collected some information from which is published data from inside the Nanda Devi National Park. I also went to Gangotri National Park near Gomukh and all other areas to find out GPS location where my animal lives. But every places I collected GPS location wherever I see fresh fecal samples. But then my species is also interesting one because it's distributed from Pakistan to Myanmar, really large. So I cannot as a single person go everywhere. So I collected GPS location where people have deposited those location in several networks such as MANIS, Mammal Network Information System, as well as people also very commonly use Global Biodiversity Information Facility called GBIF to get such GPS data. So this is a map where my animal is distributed. So I primarily found out locations where I have seen those animals in Uttarakhand, where the square red box is, whereas each of the small black dots, each of those dots I collected from the secondary data, either can be published data or from different repository. One thing people have to be very careful that sometimes GPS data can be wrong. So you have to put each of the GPS data in Google Earth and figure out if that belongs to a habitat which looks like a pika habitat or not. And when I had these information together, I use a software called Maxent with the satellite image and GPS location, which gives me quite colorful graphs. Now I'll explain what these graphs are. So see here our habitat suitability means how suitable their niche, where the animal actually lives and operates, interact with its environment. Darker color means high, green is moderate, and the lighter color is very low. So 2010, my model indicates the very darker color in Himachal and Uttarakhand, also some areas in Nepal, close to Eastern Himalaya, whereas other areas in, like for example, in Ladakh is primarily less suitable because we also found precipitation is one of the primary driver for this animal because it feeds on those plants which needs lot of water. But by 2050, when we make the predictive model, we found out eastern part of the distribution range, which is top of Arunachal Pradesh, there will be a lot of areas probably be unsuitable because it's predicted that amount of precipitation pattern will change there. Similarly, there will be changes. We found changes in central Nepal and some areas in Kashmir and Himachal Pradesh. But then see the darker color in Uttarakhand and Himachal, certain areas. These areas still will hold some amount of information, some amount of habitat, and this information can be used by different park managers, conservation practitioners to follow up that they can actually identify different pika populations and use pika as an indicator species in those areas. So for example, I know that they might die in certain areas, so I can pass on this information from my research to the conservation manager, forest department. They can look that, okay, pikas, how they're doing there. And for multiple years, they can follow up to understand if climate change is adversely affecting the native fauna or not. One of the last one I'd like to discuss is telemetry. So here in the pictures, you can see these are the collars. These are like one can put in on dogs. But then these collars really have special purposes, and people put that into lions, such a big animal, to tortoise or turtle, as well as small monarch butterfly in below in that particular picture. And to why people do it? Because these collars gives us signals or the information which helps us to understand home range size and movement patterns of this animal, to help us to understand the interaction between individuals. How telemetry works. So first, people put capture individual, put a collar. Now the collar are extra burden on the animal. So general rule of thumb is their collar weight have to be less than 5% of their body weight. And those collars with this transmitter emit signals which get recorded by the satellite or the antenna. Now collars can be very different kind of. One collar, the satellite collar, is very expensive, like $4,000 to $7,000 per collar, whereas some are like which can be detected by the antenna. Those are like $300 to $400, and some collars known as PTT collars, where they do not have battery, they are very light, but every time those animal with those PTT collars moves between the close to the receiver, they record their presence. So now all this information get collected and stored in a receiver, and they get transferred to a computer where you can visualize the movement pattern of the animal. Here is a story of a satellite tracking of Amur falcons in Nagaland, India. So Amur falcons are raptors, and they comes in like thousands in numbers in Nagaland every year, and in a particular village. So this particular adult in the picture, known as, people give them the adult as name as Naga. It's an adult male, and they captured that animal. They put a satellite collar on it, because these animals are also known to, or these birds are also known to migrate large geographical distance. So they are very curious where this animal actually go with the help of satellite tracking. So here is the data from 2013. So they, after tagging the animal, they release the animal from Nagaland, then it moves to Bangladesh, and through a Bay of Bengal, then they comes to India again, and through Arabian Sea, it reaches the African coast, Somalia, and then reach Kenya in 5 days and 10 hours time, and it traveled 5,600 kilometers. And in Kenya, then it stayed 30 days, and later it moves 100 kilometers south of Kenya to another country called Botswana, and where you can see they moved a lot. So each of these small dots are location on a particular day. So during their stay, they moved between different places. They moved in different grasslands to different landscape to find out food. Now we all know primarily animal moves to migrate because of the resources. So these animals moves from northern hemisphere, which is India, we lives in north, to the southern hemisphere, crossing the equator. When in the southern hemisphere it is summer, so they primarily feeds on rats and lots of insects. So because it is summer in southern hemisphere, that is they have abundant food there, whereas those insects are little less in Nagaland in summer. Sorry, in when they started their migration in winter. Therefore, by the end of the summer in Botswana, Zimbabwe, and Kenya, they again migrate back to India. The bigger question is, do they follow the same path? Yeah, people found almost they follow the same path, But then sometimes depend on the wind, depend on the lot of other environmental factors, there will be some variations on their path, but then primarily it's remained the same. And here are some other tools I'll just mention very quickly. This is camera trap. So camera trap is one of the very commonly used tools. Here is one of the snow leopard, which is very elusive, very difficult to see in high altitude Himalaya. So this camera is a very funny picture taken by a national, I think, Nature Conservation Foundation people. So the snow leopard is looking at one of the camera trap because it's very curious. It did not know there is another camera trap opposite of it, which actually took its picture. So camera trap pictures are commonly used to estimate population. You all know that we have tiger numbers coming up every year. So tiger numbers come up with the help of camera trap. Earlier in 1980s, the number is to comes as a pugmark, which is often not very precise. But the camera trap helps us to identify each individual based on their coat color and the patterns. So they are extremely good not only to identify the numbers of animals, but also help us to understand habitat used by different animals. Which animal actually use the habitat in the morning? Which animal use the habitat in the night? Often, if you go there, animal will get to see you and they will not come, so you're going to miss out the data. But the camera trap will give you those data, which is very exclusive. Another one of the common tool people use for ecological work is bioacoustics. And those are primarily, this is a receiver and a recorder. And people primarily record different songs, sounds from nature. For example, here below this black color picture is how a song look like. So each birds, each animals have different syllables. The patterns of their songs are very, very different and distinct. Sometimes you can identify species based on the pattern of their songs. Sometimes you can identify individual based on their songs. So these information have immense impact on the ecological study, as well as sometimes people use this information to study evolution also. So I guess I am the end of my presentation now, and I'll be more than happy to take any question if there is any.
Thanks a lot, Dr. Bhattacharya. Really a
Welcome.
illuminating talk. Really an illuminating talk, and you showed very fascinating pictures.
Thank you so much.
And very fascinating, and you make us understand very nicely. Now, this session is open for questionnaires. Okay. Okay, one question from Dr. Arnob Basu. He wants to know, please recommend an easy software from which I can get or draw digital map of our study area.
Okay, so if does he want to make it? What is the question? He wants to make a digital map of the study area. So if he did, so see, I'm not a GIS expert, but I work with collaboration. But I know one of the trouble with GIS softwares is most of them are paid. But the QGIS is free software where you can have lot of tutorials available online in either YouTube as well as in Google. There is a forum which actually tells you if you have GPS locations. So you can make shape files, which is the boundary of your study area. You can make those. Often people also make study areas, making polygons in Google Earth. But then if you want to make a really nice looking map for your publication, for your report, and if you do not have extra money to buy and expertise to buy these GIS software, such as ArcGIS, you can actually go for QGIS, which is freely available. You can use both in Mac as well as in Windows environment, QGIS.
Okay.
Also, people now use R. Sorry. Just to add, now people use R, which is primarily people use for statistical analysis. But then R also have certain packages which can help you to make maps. I have not used it yet, but then you can also try.
Okay. I think he has got his answer. Not more question I'm finding here. Okay,
So can I give some
presentation was so
clarification?
nice. I think your presentation was so nice that everyone understood.
No, I
that.
think probably people do not have much time to actually think. I might be a little faster. So another thing I'd like to say to students, because I assume there will be a lot of students. So I never thought that I'll become a wildlife biologist, right? But then my teachers were really, really nice. So they actually at Vishwabharati Shantiniketan, where I did my master's, we had certain sections where we have to go out and see how animals behave, document them. And those gives me some interest. But then when you read and when you actually try to understand certain concept, doesn't matter it's ecology or evolution or genetics or whatever Dr. Chottara says about endocrinology, there is a huge amount of resources available in YouTube, which is freely available. Facebook is very, very important to connect with our friends. Also, there are Facebook groups which can give us some information. So those information you can actually help to make your understanding better. And a lot of things which show in Discovery and National Geographic are often not the reality. Reality is much more harsher, and only you understand when you go to field and do the field work. So not everything is so rosy projected by those movies. So I hope that these give my presentation gives some kind of idea why people use certain tools for ecological research. And these are some commonly used tools, but people use a lot of different tools because with the advancement of different kind of techniques we are having now, every day there's something new coming up.
Exactly. Okay. Really, it's a hectic job in the field of the research of ecological sciences. Really, it's a hectic.
Atri, I'll probably correct you. So if you write, say, hectic,
also
it will probably have some negative in a connotation.
Fascinating.
You have to be,
as well.
Yeah, you have to be interested. It can be challenging. It's a politically correct way of saying. It is a challenging job. Hectic includes that probably you will not able to balance it out. We just have to have to positive information to our students. So it's a challenging job. You have to be interested to actually to find out something. And there is nothing wrong that if you feel that ecology might not be a field, you might want to do something else. Because none of us, Atri or Dr. Chottaraj, we all learn through our experiences. So for the students who are thinking research can be a career, so it's it's a probably word of wisdom from us who did PhD that it's okay not to actually be very sure that what you want to do, but then just to be honest to yourself.
Yeah, truly.
Thank you, Atri, for giving me the opportunity.
Thanks a lot, Dr. Bhattacharya, and we hope in future we will get you for more things to discuss. Okay. Thanks.
Thank you so much. Yeah. Take care. Bye. Have a good day.
Bye. Okay. Now this session is at its end. It is the valedictory session, and vote of thanks will be given by my colleague, Mrs. Shatarupa Pal. Shatarupa, please, it's over to you.
Thank you, Atridi. First of all, very good afternoon to all of you. Honorable dignitaries, respected guests, dear participants, and our beloved students. On behalf of our Michael Madhusudhan Memorial College, its entire fraternity and organizing committee, I feel immense pleasure to take the opportunity to propose the vote of thanks. Firstly, I would like to express my sincere gratitude to our Honorable Principal Sir, Dr. G.M. Helaluddin, for providing us the opportunity to organize this webinar. Without his encouragement and guidance, this webinar would not have been possible. We are extremely grateful To our respected eminent resource person, Dr. Oshumanto Chattoraj and Dr. Shobhuj Bhattacharya were able to spare time from their busy schedule to grace our webinar and dispense their knowledge. The discourse of Dr. Chattoraj challenges the contemporary paradigm that modern artificial light are technologically marvels that have no negative side effects. I'm sure that most of our participants were unaware of this novel research field investigating the effect of light pollution caused by high energy artificial light on our daily life and overall health. Thank you so much, sir, for enlightening us by your remarkable research findings. In his address, Dr. Shobhuj Bhattacharya highlighted the use of cutting-edge technologies in the ecological research field that has not only enlightened our participants, but is sure to have excited and inspired them. Thank you so much, sir. Then I take the opportunity to specially express my deep regards to our respected and beloved head of the department, Dr. Atreyi Shahana, for her unwavering support and confidence in us. My deep sense of appreciation to all of our participants who chose to be live with us and attend the webinar with great enthusiasm and have made it a successful event. My deep gratitude also goes to the organizing committee members for selecting such relevant as well as captivating topics. I think Mr. Shadhin Kumar Mondol, Assistant Professor, Department of Sanskrit, Michael Madhusudan College, for technical support and helping us in webcasting the webinar through YouTube channel. Last but surely not the least, students of our department and their heartfelt desire made us very enthusiastic for organizing this webinar. Once again, I thank you all of you for being with us this afternoon. Have a wonderful day ahead. Over to you, Atreyi.
Okay, thank you, Shatarupa. Thanks a lot. It's my pleasure, rather, that you took the responsibility to give this vote of thanks. Thanks a lot. Thanks for your sincere support. Now I have to say a formal goodbye to all of you. Thanks a lot for your patience, and we hope in future we will get you all for our next and further webinars what we will going to conduct. Okay, thank you. Thanks a lot.